Hello everyone, I welcome you all to a video on CRISPR, which is a very interesting and comparatively new technology. The full form of CRISPR is clustered regularly interspaced short palindromic repeats. Its name, in itself giving some idea about the structure of the CRISPR. These are the short repeats which are palindromic in nature. Palindromic means they are same in both the direction. If you read the sequence, then the sequence is going to be same forward and backward. CRISPR is a remarkable and revolutionary tool for gene editing. If you know about the gene editing, it's a technique or say a group of techniques. It's a broad term to change or alter the organism's DNA. With gene editing, the short genetic sequences can be deleted or they can be added or even modified at a particular locus in the genome. Although there are many other techniques also available for the gene editing like gene zinc finger, meganucleases, but CRISPR is the most recent one and probably the best one. CRISPR is naturally found in archaea and bacteria, where it plays important role in protecting the bacterial cell from invading viruses or bacteriophages by cleaving the foreign DNA. That means by cleaving the DNA of bacteriophages. It is the part of defense system of bacterial cell, and if you see its features, they are somewhat similar to our adaptive immune system. It is, kind of surprising that these simple prokaryotes have such a complex immune system, which even have memory response. Memory response is important characteristic of the adaptive immunity, and here that kind of memory response is present in bacteria. So that is what it is surprising. That means if the infection has occurred in the past, it will be remembered by the bacterial cell, and next time or the second time, whenever the cell is invaded by the bacteriophage, the immune response would be more quick and more efficient. So that kind of memory response is there. It was discovered in 1987, but in 2007 the function was demonstrated in Streptococcus thermophilus. It was noted that whenever a new bacteriophage attacks the bacteria, a new spacer sequence was incorporated. And another noticeable thing was that the DNA sequence of a spacer was same as part of the viral genome. So that showed that the bacterial cell keeps the record of the infection in the form of a spacer. We'll discuss these spacers in length. Let's see how CRISPR looks like. Here you can see the CRISPR locus. You can also call it CRISPR array. The two characteristic feature of CRISPR are that there are repeats, which I have shown by black diamonds, and also the second component is a spacer. So spacers are these oval shaped structures which I have shown colored. Why I have shown these in different colors? Because all these spacers are different, and all these spacers are having the memory of the bacteriophages which have attacked to this bacterial cell in the past. Here suppose 3 different spacers are shown, so that means at least 3 bacteriophages have attacked this bacterial cell in the past. This kind of 50 units are possible in one of the CRISPR locus. The CRISPR repeats, these black ones, they may be around 28 to 37 base pair long, but it may be shorter and longer also. And the spacer sequences can be 32 to 38 base pair, although they may range from 21 to 72 base pairs. And as we have already discussed that these spacers are, they have the memory of the past infections. So you can say that this CRISPR, it's a kind of a storehouse for the for the enemy's record. It's a kind of library where you store or you record all the sequences, all the record of the previous infections which have occurred. So this is, quite interesting. And then you can see the leader sequence. This is 80 rich. And here you can see adjacent to leader sequence, there are Cas genes. Cas is C for CRISPR and AS is for associated. So that means CRISPR associated genes. These CRISPR associated genes, they form different type of Cas proteins like Cas1, Cas2, Cas9, etc. Out of these, Cas1 and Cas2, these are universally present in all types of CRISPR-Cas system. Actually, there are three different type of CRISPR-Cas systems which are known as CRISPR-1, CRISPR-Cas system 1, 2 and 3. Out of these three, the second system, that is more common one, and Cas9 is present in that only, and that is the most studied one also. So we generally talk about the second system. Then here also you see that CRRNA is formed by the spacers, and means according to different colors of spacers, I have shown the colors, different colored CRISPR RNA also, CRRNA which is known as. And then at last you can see there is a tracer gene. This tracer gene naturally forms the tracer RNA. So this is the structure of CRISPR. Now this one is the spacer acquisition. That means how a new spacer is added to the cell, to the CRISPR locus. So this whole thing is exactly similar to this one. So that's why I have not named it here, but it is exactly same. Now you see what is different here. The bacteriophage is attacking. So this is the bacteriophage which is attacking this bacterial cell for the first time. What it will do? The protein coat is left outside, only the nucleic acid is inserted into the cell. This is the enlarged view of the nucleic acid. In this you can see there are two sequences, and these are PAM and protospacer. PAM is protospacer adjacent motif, very small 2 to 7 nucleotide long sequence, but very important one. And Cas1 and Cas2, these are the two enzymes or two proteins which recognize the invading DNA. This is the invading DNA, this is the invading bacteriophage, this is invading DNA. So Cas1 and Cas2, they recognize this invading DNA and they form a complex. They form a complex which is required for this insertion process. And spacer acquisition is PAM dependent. So PAM should be there in this in this nucleotide sequence. But though PAM is very important here, but PAM is not inserted into the CRISPR locus. Only this protospacer is present here. Only this is the acquisition of the protospacer takes place, and now it is called as the spacer. So this is yellow colored protospacer, that's why I have shown this is spacer by yellow color. So only this spacer is added here or inserted here, not the PAM. PAM is cleaved before protospacer is incorporated here. So that's how a new spacer has been incorporated in the CRISPR locus. See what happens next. Let's now discuss the components of CRISPR. It has basically two components. First one is the guide RNA. G is for guide. So it is non-coding short RNA and it has a spacer sequence. guide RNA is made up of CRRNA. Here you can see at 5' end CRRNA, and this one is the tracer RNA. So these two components are there. These are generated by two different genes, as we have just discussed, that CRRNA is formed by CR genes or the spacers, and the tracer RNA is formed by the tracer gene. Tracer RNA has secondary structure, and that's why you can see this stem loop like structure here, and this kind of structure, secondary structure, is not present in CRRNA. CRRNA, although, is target specific sequence because it is produced from the spacer. So and there could be two separate RNA molecules, that is CRRNA and tracer RNA, as they are shown here. These are two separate molecules. Or alternatively, there can be single gRNA. gRNA means guide RNA. So one 5' end, one 3' end, and here you see those two sequences, those two RNAs, they have been included in single RNA. So this kind of single gRNA can also be formed. Actually, this is more common. And yeah, one more thing, that there is some portion of CRISPR repeat also present in the CRRNA. In this portion, in the flanking regions, these are the flanking regions. So at that point, a little CRISPR repeats are also present. This guide RNA is found naturally in bacterial cell, but whenever needed we can design it as per the demand, and these are these days synthesized artificially in lab. Then there is second component, which is known as Cas9 protein. This is CRISPR associated protein 9. It acts like endonuclease enzyme. What is the function? The function of Cas9 is that it unwinds the foreign DNA. Foreign DNA means the invading bacteriophage DNA. And it looks for complementary region to spacer, and if it finds the complementary region, the Cas9 cleaves the invading sequence of bacteriophage. So that is what its function is. Let's now discuss mechanism. Here you see CRISPR locus. This is the CRISPR locus, and the tracer gene and the Cas genes. So this is exactly same diagram which we have discussed before, and I have shown this spacer from yellow color, which is the one, if you remember, the new spacer which has been incorporated into the CRISPR locus. Now here you can see from this CRISPR locus the CRRNA is formed. This single-stranded CRRNA is complementary to the bacteriophage, and this CRRNA actually gives rise to single guide RNA. Guide RNA now gets attached to the Cas9 protein. Here you can see the complex is formed, and this complex is now known as sgRNA Cas9 complex. Guide RNA then guides the Cas9 to recognize the target site in the bacteriophage DNA. Now here you see there is a bacteriophage, the same bacteriophage, but now it is attacking the bacterial cell second time. So when it attacks the bacteria second time, again the nucleic acid or the DNA is left inside the cell. You see the protospacer and also the PAM sequence, and then you see what happens. So this Cas9 binds with the PAM, it unwinds it. So what it does, this is the invading DNA, the Cas9, it is doing the unwinding. Now when it unwinds, what will happen? It already has this protospacer and PAM sequence. This whole sequence which I have shown by red, this is the sequence of this bacteriophage, which is having protospacer and PAM. Here you can see protospacer and PAM. Here also you can see protospacer and PAM. So after the unbinding, what will happen? This guide RNA is already there, and this guide RNA will bind to this protospacer. Why it will bind? It will bind because these are complementary sequences. So when they bind, what they will do? They will, cleave both the strands, and the double-stranded break will be formed at these points, which I have shown here by the red colored arrows. So at these points, which are adjacent to PAM, at this particular point and at this particular point, the DNA will be cleaved. Let's see how this RNA and DNA duplex is cleaving the sequence. Yeah, here you see. This was the target, this was the bacteriophage DNA. This is the PAM sequence, and here you can see the double-stranded break, and the cleavage has occurred here, and this is Cas9 mediated cleavage. One thing I must tell you here, that the PAM are required to degrade the invading DNA. PAM actually distinguishes self and non-self DNA. Since PAM is not present in CRISPR locus, so they are not degraded. Only what is degraded? Only this foreign DNA is degraded. Why this foreign DNA is degraded? Because this PAM is there and PAM is recognized. After the cleavage of both the strands, the natural repair system of the cell is activated, because in every cell there is a repair mechanism, and this is now activated. There is a gap, and this gap has to be fixed. That is the natural process. All the cells have two types of repair systems, or you can say two types of pathways. These are known as non-homologous AND joining, commonly known as NHEJ, or homology directed repair, that is known as HDR. So these two types of repair systems are present in all cells. Normally both are present in the cell, both work together, but remember this thing, that both are error prone. Whenever the cell goes through these mechanisms, these repair systems, the errors take place. The first one, which is NHEJ, it repairs the AND without homologous template, because it is non-homologous AND joining. But it can introduce errors like insertion can take place, deletion or modification. These type of errors can take place. Whereas this second one is the homology directed repair, which is commonly known as HDR. It is a kind of homologous repair. So here the gap which was made by double-stranded break is fixed by the short sequence of DNA. So you need a short sequence of DNA here. That means here a short strand of DNA is required as a template, and it leads to what? It leads to knocked in sequence of interest. For targeted gene editing, means when we use this technology for our own benefit, or say using it in its one of the applications, the double-stranded breaks are repaired by second pathway, that is HDR. So we always prefer HDR pathway when we are doing the targeted genomics. In this figure, we'll discuss how CRISPR technology can be used for gene correction or any other applications. First, the target DNA is identified. So here you can see the target DNA. CRISPR is a remarkable technology in the sense it can be used to cut any region, and any region means literally any region of DNA in any of the organism can be cut. That is why CRISPR has innumerable applications. And how it can be done? It can be done by simply changing the nucleotide sequence of CRRNA. You know how the CRRNA was formed? So if you just change the nucleotide sequence of cRNA, you can, change many things. So the scientists, what they do, they synthesize the specially designed cRNA which acts like guide molecule. So here you can see this is cRNA, which is actually the guide molecule, and this is the specially designed one. So scientists have actually specially designed it according to their need. This you can call as the editing molecule. This molecule is introduced in the cell by viral or the plasmid vector, or if you are not using any vector, then by direct delivery methods also, like electroporation, etc. These can be introduced into the cell. And this is around 20 nucleotide long stretch, which should be complementary to target DNA. While designing this editing molecule, it should be kept in mind that this 20 nucleotide segment should be unique to this target gene. Target gene means the gene in which we want to do the editing. So this is the target gene or target DNA. So the sequence of that 20 nucleotide, that should be very unique to this, and it should not be present in the genome, because if it is present in the genome, what will happen? It will cleave the genome at all those points also, and that would be kind of disastrous for the cell. Now you see what happens. The cRNA or the guide RNA along with the protein, it can act upon the target DNA, and the double-stranded break is formed. This mechanism we have already discussed. Now, what next? Once the break is there, it will go through the repair system, and when doing the manipulations or the gene editing, we try to get it done through HDR. Remember, I told you that the HDR method is better for this. And the cell should go through HDR. For that, we have to do, somehow we have to inhibit the other pathway, that is NHEJ pathway. There is a key enzyme in NHEJ pathway that is known as DNA ligase 4, and one way of inhibiting this NHEJ pathway is to inhibit the enzyme. But then there are many other ways also. Any of these ways can be used so that the HDR pathway is encouraged. Then what we do? Then a customized gene sequence is synthesized. This customized gene sequence, which I have shown by yellow color, here you can see this yellow colored is the customized gene sequence, and at both the ends there is a sequence which is homologous sequence. So this green colored sequence is homologous to this particular one. That means in the target DNA there was some region that was included in customized gene sequence. So here you can see this is the one which we have synthesized, and this has been synthesized so that the homologous recombination can take place. So here you can see these cross, these are showing that the homologous recombination is taking place at this point. And then next, the insertion of this sequence takes place and the gene is kind of corrected. So that is the genome editing. Or because here we are using the gene sequence which we want to insert, so that's why, and we are inserting it in the target DNA at a point where we want to insert it, so that's why it is known as targeted genome editing. With CRISPR, multiple gene targeting can also be done, which is something very beneficial and makes this technique suitable and very convenient for many applications. CRISPR has unlimited applications. Here I will discuss just a few of them, and that too in short. Many researches are going on to modify the animals. Livestock with disease resistance have been produced with the CRISPR technology. Besides, genetically modified salmon fish that grows very fast and is more nutritious has already been produced in Canada. Recently, researchers at UK proved that a gene which stops female mosquitoes from laying eggs can be introduced in mosquitoes that spread the malaria. This may be a way to control the malaria all over the world. Only future will tell whether we'll be able to control the malaria by CRISPR technology or not. Pigs can be modified to make its organ suitable for the transplant in humans, and if that can be done, it would be really a boon for the human beings. This technology can make the pet owners very happy because now the pets can be custom designed for colors, coat pattern, and desirable traits. So amazing, isn't it? CRISPR technology can do miracles in therapeutics. Experiments and trials are going on to treat cancer, HIV, and hepatitis, and many other diseases also. It can also cure even the genetic diseases. Similarly, genetically modified crops for stress resistance, herbicide resistance, drought resistance, disease resistance can be produced. The crops with improved nutrition and improved productivity and yield can also be produced by CRISPR technology. So there are actually endless possibilities of CRISPR, but for today that is enough. Hope you found this technology interesting. We'll meet again in another video. So bye for now, and your comments are welcome.